Okay, good. So welcome everyone. This is actually the first of several of the different types of meetings that we're going to have, different types of presentations. As you know, in the context of MOHCCN, we had various requests for applications, various competitions. Some were salary awards, some were grants. And as we're the term of these grants or awards is coming to an end, and as of course phase one is coming to an end, and as we're thinking whether or not we should renew some of these programs, we think it's very important, and it's going to be important also for our reporting to Health Canada. It's important to see what people have done. So this is the first of these things. So this is one is about health informatics and data science awards. So some of the awards were given to master student, other to PhD students, and other to postdocs. So today they're going to tell us very short, very briefly about their presentation, and maybe Veronique is going to tell us a bit more about how much time they have and when are going to be the questions.
Perfect. Thank you so much, Andre. Welcome everyone to this really exciting new series. As Andre was saying, these are going to highlight awardees from the network's Health Informatics and Data Science competition. So since we do have a little bit of catching up to do, we're actually going to be hearing today from awardees from the first two rounds of the competition, which were awarded in 2023 and 2024. These were one-year projects meant to use network data to advance high-quality precision oncology research. The award was designed, like Andre said, to support young researchers during graduate or postdoctoral studies. And some of the speakers you'll hear today, because as I said, we have a bit of catching up to do, have actually completed their studies and launched their career since the award has ended. So it's a great mix of people that we'll be hearing about today, or hearing from today, excuse me. The other thing I want to highlight about this seminar is that it's a little bit different from ones that you might have attended network seminars in the past. So as Andre said, the presenters were asked to prepare 3-minute presentations using just a single slide and to share their work in plain language. So this is kind of similar to a 3-minute thesis competition, although there won't be any judging going on today. Another unique aspect of this series is that it also does involve an element of training in a way. So 2 weeks ago, the presenters that you'll hear from today met with some generous volunteers from the network's patient working group to practice their presentation. I sat in that meeting and I can tell you that the patient working group members had really thoughtful and constructive feedback to give the presenters that they were asked to incorporate into their presentation today. We really think that this is a great opportunity to bring network trainees together with people with lived experience to learn from each other and to help these early career researchers develop their skills in sharing their research with a broad audience. So I do want to give a huge shout out to Debbie, Kirsten, Jennifer, and Lou for participating in this round. I'll also, sorry, yes, Andre.
Can I make a comment about this? I think this is like a very rich experience that the patients are allowing our trainees to have. I think, getting genuine and sincere feedback from patients who are, of course, they know a lot about cancer, but they're not scientists, or most of them are not scientists. So I think this is like very privileged. And it's something that I think you should always remember when you talk about your science later on, that you're not talking to scientists most of the time. You're talking to lay people and basically people with lived experience. They're pros, but this is obviously something always important. And I think you probably had a lot of learning from this experience with the patients.
Thank you, Andre. Those are really great points. And I think it's really important to highlight, all the work that's gone into this particular series. So thank you for that. I will also just do a tiny bit of housekeeping before we move forward. We will be keeping questions until the end. We're going to go through all the presentations first. Please either type your questions in the Q&A box, which you can find at the top of your screen. It's 2 little speech bubbles with a question mark. Or at the end during the question period, you can raise your hand and we'll allow you to unmute and then you can ask them live. I also want to take a quick moment to acknowledge that I personally, as well as some of our speakers today, are calling in from Mi'kmaq, the traditional and unceded territory of the Mi'kmaq people. And I want to acknowledge that our speakers and attendees today are located all across the country on lands that are home to diverse First Nations, Métis, and Inuit populations. And I invite everyone here to recognize that, to honor those peoples, and to reflect on the importance of those lands and waters and peoples. Finally, I have the great pleasure of introducing my co-chair, Debbie. Debbie is one of the inaugural members of the network's patient working group. She is also a passionate advocate for the network. On top of all the work that Debbie and all of the patient working group are doing, Debbie generously volunteered to co-chair her second network seminar and to join the pre-meeting with the presenters a couple of weeks ago. So thank you so much for joining us, Debbie, and I'll pass it to you to get us started.
Thank you, Veronique, and I'm delighted. I'm honored to have the opportunity to be here today to introduce the next generation of experts who will help make precision oncology a reality to patients. I'm a breast cancer survivor, registered nurse for 37 years, and caregiver to my mom during her cancer journey 2 years after my diagnosis. And at the age of 42, I was diagnosed with locally advanced breast cancer. It was estrogen and progesterone positive, HER2 negative. The tumor was a large tumor the size of a lemon, and I had metastases in 3 lymph nodes. A 9-month journey was inclusive of radical mastectomy. 36 lymph nodes were removed, and I had 25 rounds of radiation. This was a critical turning point in my life, and I developed a new resilience and courage to face each day with a heart full of gratitude. I grew my understanding and appreciation for the critical role that research played in saving my life and enabling me to be here today to speak and to share my story. This research will enable precision oncology cancer care advancement. It will give hope, and it will fulfill the goal of a cure for all cancers and save lives. To this point, I'm so pleased to have the opportunity to interact with and help advise trainees funded through the network. As a patient, I think it's very important that researchers, especially ones early in their careers, have opportunity to hone their communication skills with different audiences. Like Veronique said, about 2 weeks ago, all 5 speakers you're about to hear from today met with me and 3 other members of the patient working group to practice their presentation. We gave them feedback highlighting what aspects of their presentation worked well and gave them advice on how they could improve their slides and their delivery. We had some great interactions, and I hope the presenters found it as valuable as I did. I'm excited to see their presentations again today. Without further ado, we'll get started with our first presentation from Ali Khalijali Merabadi. Ali is a PhD candidate in the AI in Medicine lab at the University of British Columbia. He'll tell us about his work developing an AI search engine for diagnosis of primary and metastatic cancers. Thank you, Ali, and we appreciate you sharing this informative presentation now.
Thank you for your kind introduction. I'm happy to be here and present my work. When cancer spreads, finding where it started can be a challenging mystery even for pathologists and experts. We can have a tumor sample from brain, lymph nodes, liver, or bones, but it can actually be a lung cancer, where the cancer originates from. So this place that the cancer starts is called primary site of origin in metastatic cancers. And as part of clinical practice, we generate routine pathology slides, which are basically a small tissue, a small tumor sample for pathologists to identify the cancer subtype or look for certain pattern. But what pathologists can't reliably do with these slides is to identify the primary site of origin. And for that, they usually request further testing like molecular or genomics. So here our question was that, can we use the power of AI here to identify the primary site of origin using these tissue slides or not? And whether AI can identify the metastatic status just using these slides. So the way that the AI model can answer this is like a Google search. Think of it like a Google search. The AI model goes, and when new patient comes into the clinic, goes to the hospital archive and finds all the similar patient in our hospital's cohort to those of the new patient. Here, the red and blue groups of patients are our matching groups of patients. By having these matching groups of patients, we can actually learn from what happened to them to make decisions for these new groups of patients, especially for rare cancers. This can be in terms of their outcome, their treatment, and their shared biology for doctors to make a more personalized treatment based on the similarity with the previous patient. So we put this idea into test on a rare metastatic cohort from BC Cancer's POC program, which is part of Marathon of Hope. And we had more than 1,800 rare cases covering more than 14 organs and also more than 225 different cancer subtypes. So on the primary site prediction task, which is a super challenging task even for the doctor, the model could achieve 40% accuracy. This is still 6 times better than random. And also the model could differentiate between primary versus metastatic cancer with an accuracy of 80%. So given this challenging nature of this task, this is a really good performance for this model, given that we didn't use any human annotation or label for training this model. So the model is completely unsupervised and can be served as a tool for pathologists and doctors in the hospital to narrow down the search space and the possibilities for each new patient in the clinic. Thank you for this opportunity, and I'd be happy to answer any questions.
Thank you, Ali. We appreciate you sharing this very informative presentation, and we will have some Q&A at the end. Thank you. And we'll move on to our next presenter, which I will introduce now. That is Parsa Bagzurda. Dr. Parsa Bagzurda was at the time of receiving the award, a postdoctoral fellow in the Shirin Abduzanaj Enger's lab in the Department of Oncology at McGill University. He'll tell us about his work in basal cell carcinoma. Parsa, I'll turn it over to you.
Thank you, Debbie, for the introduction. Hello, everyone. So yes, this is a joint working collaboration with Dr. Enger and Dr. Philippe Lefrancois. We have worked on basal cell carcinoma patients. Why we chose this? Because it is one of the most common types of skin cancer, and usually this disease is treated outside of the oncology departments, usually in the dermatology and pathology settings. So this means that we would have some issues about the underreporting of the basal cell carcinoma cases. But pathology reports contain key informations that we can extract to enrich the cancer registries. But the manual review is very slow and error-prone. So in this project, we have focused on an automatic pipeline to first better track and extract information regarding basal cell carcinoma patients, and also to refer them for further interventions. So we have defined 2 tasks to first extract BCC tumor descriptions automatically from pathology reports. We developed a natural language processing pipeline, and then we use these extracted segments to suggest next interventions and next steps, including follow-up, re-excision, Mohs surgery, and so on. So we collected 380 7 reports from 180 patients and trained the AI model on the two aforementioned tasks. We had very promising results for the task 1, which was the identifying the textual span or the textual segments within the pathology reports. We achieved the F1 score of 90. It means that 90% of all of the mentions, basal cell carcinoma mentions within pathology reports, we could extract them successfully, and 86% of all of the referrals were correct. So this is a promising result for leveraging an automatic pipeline for the referrals for the patients. So the impact of this research is that this could enhance to populate the cancer registries, specifically for those type of skin cancers that are usually treated outside of the oncology settings. This pipeline, we hope that it would ensure that the patients would get the proper follow-up and the best decision for them. And also, we are hopeful that we can extend this pipeline to other types of skin cancers, such as melanoma or so on. Thank you for your attention.
Thank you, Parsa, for sharing that very insightful presentation as well. And we will take question and answer at the end again. Our third presentation is from Riley Arsenault. Riley Arsenault is a PhD, candidate in Jeanette Boudreau's lab in Dalhousie University, and she'll tell us about her work looking at immune cells in pancreatic cancer. Thank you, Riley, and I'll turn it over to you now.
Hi, I'm Riley. Oh, let's see if this slide. There we go. Hi, I'm Riley, and I'm going to present on my project, which is Immune Patterns Predict Longer Survival in Pancreatic Cancer. So pancreatic cancer is one of the deadliest cancers, and only about 1 in 10 people survive 5 years after diagnosis. And one reason pancreatic tumors are thought to be so deadly is because they're cold, meaning that they lack immune cells. So this is the body's defense army that normally helps fight cancer. And without those cells, the cancer can grow unchecked. But we know that some pancreatic tumors do have immune cells, so we asked what makes the difference in survival if it's not just dependent on the presence of these cells. In many cancers, immune cells attack and kill the cancer cells, and that's why immunotherapy, or treatments that boost the immune system, have transformed care for cancers like melanoma and lung. But in pancreatic cancer, immunotherapy hasn't worked well. So we wondered if the answer lies in where the immune cells are inside the tumor and not just how many there are. And one reason we're looking at where they're located is because the immune system works like a team. So these cells don't fight alone, and they need to communicate and cooperate to launch an effective attack. But immune cells can only talk to each other if they're close enough to interact. And that's why their localization inside the tumor matters. So a scattered team can't win the game, but an organized one can. So we analyzed tumors from 73 patients with pancreatic cancer using advanced imaging and computer algorithms. And think of it like creating a map of each tumor. So we pinpoint the location of every immune cell and then saw how they clustered. And we looked at several immune cell types, but 3 became especially important. One was killer T cells, so the soldiers of the immune system, which can kill cancer cells. The next was helper T cells, or the coordinators. These are generals which organize and direct the attack. And then there were B cells, which help equip and activate T cells and support the fight. So when we looked at where these and how these immune cells grouped around the cancer, we found that patients with more helper T cells, or those coordinators, near the edges of the tumor lived longer. And these helper T cells often appeared alongside killer T cells and B cells, forming what we called a T cell neighborhood. So patients with more of this T cell neighborhood have had better survival, whereas other immune cell clusters didn't provide the same benefit. And our study shows 2 things. So the first is that pancreatic tumors aren't all the same. And the second is about when immune cells gather and interact in organized patterns, patients tend to do better. So understanding these patterns could help doctors predict survival outcomes and potentially identify who might respond to immunotherapy. So in short, mapping the body's defense army and understanding how its members work together could help turn a cold tumor into one the immune system can fight. Thank you.
Thank you, Riley, for that excellent presentation. We appreciate you sharing, and we will move on now to our fourth presentation, which will be from Peter Herr. Peter Herr is a PhD candidate co-supervised by Hansen He and Benjamin Halib Khan's in the Department of Medical Biophysics at the University of Toronto. He'll tell us about his work looking at circular RNAs across cancer types. So Peter, I will turn it over to you. Thank you.
Thank you. So yes, today I'll be talking about my work on circular RNAs. So while doctors review tests and give their best predictions, there's still a major challenge where two people with identical looking tumors can have completely different outcomes. This is because often we're missing information like critical clues hiding inside cancer cells that we aren't measuring. And hidden in our cells are molecules called circular RNAs. And in the figure, you can see how, unlike regular genetic material that forms a straight line that gets degraded after the cell is done using it, these molecules loop back on themselves into a closed circle, and this shape makes them very stable. And this stability also makes them potentially useful for tracking cancer, since they can be detected in the blood and urine. However, most cancer research programs disregard them as standard laboratory methods exclude them. So the world's largest cancer databases can't tell us anything about them. The Marathon of Hope Cancer Centers Network is different by using specialized methods that preserve these molecules, giving us a unique window into Canadian cancer patients that other programs don't have. So to this end, I'm building computational tools to profile for circular RNAs across MOHCCN patient samples. So I start off by analyzing data from each tumor, searching for a specific feature which differentiates circular RNAs. And then I measure how much of each circular RNA every patient produces and create a profile unique to each person. We then look for patterns. So do these patients naturally cluster into distinct groups? Do certain patterns predict who will respond to treatment and who won't? And crucially, do these molecules tell us something that conventional tests miss? So early results suggest they do. And in some cancer types, we found that patients with different circular RNA levels had higher risk of their cancer returning. Along with this, we're curating a catalog of circular RNAs for clinicians to be able to access and query for further studies. And that's information that could be helpful for treatment decisions. And because these molecules are so stable, we might eventually detect them in a simple blood test, so no invasive biopsy, is required. To this end, the goal is to give clinicians new tools to answer the question of how a person will respond and translating this into personalized predictions. Thank you.
Thank you, Peter, for sharing your informative presentation as well. And we will move now on to our fifth and final presenter today, who is Michael Gunish. Michael Gunish is a PhD candidate at the Kieran Campbell's lab at the University of Toronto. He will tell us about his work looking at immune cell organization in tumors. I will turn it over to you. Thank you, Michael.
All right, thank you for the introduction, Debbie. I'm going to talk a bit about immune system cancer interactions as well and how these can change in 2D space. So it's been known for a long time that more immune involvement is good for patients, right? So on the top left-hand side, you see the overall survival probability of 10,000 different patients across over 15 different tumor types. And as you can tell, patients with lots of immune involvement survive longer than patients with fewer immune involvement. One of the reasons this works is because of an immune cell type called a killer T cell. And this T cell is constantly on the lookout for dangers, including cancer cells. And it does this based on a receptor that is on its outside that matches a protein complex encoded by the HLA genes. And if these two match, then a killer T cell can detect the cancer cell and kill it and essentially get rid of this danger. In the best case scenario, a patient might start developing cancer, killer T cells identify this, get rid of it, and the patient sort of never notices because it never becomes a clinical problem. But cancer also mutates over time, and so it can acquire mutations in these HLA genes that essentially allow the cancer cells to hide from the immune system. So the T cells are still there, they're still looking for things, but they can't find the cancer anymore. So that's what an HLA loss would do. It's kind of like a cloak of invisibility for the cancer cells. And so what I've done is I've mapped these HLA loss events across hundreds of patients in pancreatic cancer and identified that this happens in about 26% of cases. And then finally, because we also have spatially resolved data, meaning we know where immune cells are located in 2D space and where tumor cells are located in 2D space. I calculated the distance between killer T cells, for example, and tumor cells. And that's what you see on the bottom left plot. So this is essentially calculating the distance between those cell types and looking at how this changes with an HLA loss. So in male patients, for example, you see a slight decrease in distance. So the killer T cells are closer to the tumor with HLA loss, while in females, you see the exact opposite. I further quantified this using the slopes of the lines you see. And then obviously we don't just care about killer T cells, we care about lots of different immune cell types. And so I've repeated this analysis for every single cell type that we've measured. And as you can see, if the slope is positive, this cell type is further away from the tumor, while if the slope is negative, it is closer to the tumor. And this is almost a perfect mirror image between males and females. So whenever a cell type is closer in males, it is further away in females and vice versa. And so the hope is that this information will in the future allow us to better stratify patients and also predict what treatments they might respond to. Thank you.
Thank you very much, Michael. Thank you to all of our speakers. Those were fantastic presentations. And congratulations, great job on staying on time. So because of that, we actually have quite a bit of time for questions. Debbie, do you have a question to start us off?
I do. Thank you, Veronica. And once again, thank you as well for allowing me to hear all of those great presentations again. It was excellent. My first question is to Ali. I'm very interested, Ali, in the field of AI use in medicine. And do you feel that AI tools could serve the underserved communities with positive outcomes where oncology specialists may not be as easily accessed, therefore enhancing care outcomes for patients in these areas?
That's a really good question. Yes, I think that's one of the most important promise of AI in healthcare. And with that, because everything processing-wise can be happening on cloud, even remote areas or hospitals that are in very, they don't have access to pathologists, expert pathologists, they can easily verify their findings or use AI as an adjunct tool to help them.
Thank you, Ali. I was hopeful for your response in that regard, and I'm interested to learn. I'm interested to see how things progress in this area. So thank you for that.
Of course.
All right. I'll turn it back to Veronica.
Thank you so much, Debbie. Andre, I see you have your hand up.
Yes, first I have a comment and then I have a question. The comment is that these were excellent presentation with very interesting results. So I have to congratulate all of you for this great work. Also your supervisor, obviously they did a great job. And I have to say also the members of the committee who selected you. I think they selected really good people. So this is very nice to see. My question is for Michael. So Michael, this was very surprising, no? Your result, the difference in sex or gender?
Yes. Yeah, it was extremely surprising. In fact, I mean, there's a whole bunch of things that I couldn't go into that are also sex specific. It's sort of a bit of a mystery still why it's the case. There's a few potential leads, but yes, completely unexpected coming into this project.
So what would be like some, I mean, is it relate? I mean, does this relate to the types of cancer or in part, or obviously it can be different between men and women, hormonal effects? What kind of leads do you have for possible hypothesis or for hypothesis?
Yeah. So first of all, this is only in pancreatic cancer where we've looked at the sex-specific effects, right?
Right.
There's lots of different hypotheses. So for example, I mean, the two biggest differences that come up between men and women are, well, an X chromosome, an extra X chromosome and a Y chromosome in males, right? So that's one possibility, but I haven't found any sort of genes that are associated on either chromosome that would explain any of this. There are also, hormones are another good one. Unfortunately, we don't have hormonal measurements of any of these patients. So that's sort of a great guess, but I can't currently confirm or deny, essentially test that hypothesis. There is also X-linked inactivation that could happen. There's a bunch of different mechanisms, but so far nothing has really come up.
And pancreatic cancer is the same kind of disease between men and women?
For the most part, yes. So there's two main transcriptional subtypes between in pancreatic cancer in general. There's a basal and the classical subtype. The basal one happens more frequently in males, but it also happens in females. To what extent the basal subtype is different between males and females is unknown.
Okay. Good. Thank you.
Thank you. Maybe we can try Dr. Hawk. I see you also have your hand up. You should also be able to unmute yourself now.
Yes, I am. Hi. I completely agree with Andre. It's excellent. I mean, I was excited when I listened to the all of them. But I guess my question is directed mostly to Ali and Parsa. So I'm coming from the industry world, the tech transfer world, and I was wondering if either of you have you been able to implement this at your centers? I mean, is there a willingness, or is there an obstacle? So, like, how would you? It sounds to me like you're ready to go. You have the AI, you have the program, you have the proof that it works and specificity and all that. But is it being used? Is it being used in the clinic?
So, if you allow me, I would go first. So. Since our project, the REB for our project was coming from a biobank, so the main REB and the backbone for this was a biobank that we had in our hospital. So the data extraction pipeline actually was there. So part of the pipeline for extracting pathology reports and all of the related referrals that we had to extract from the system to train our model was already in place. So I did many projects during my postdoctoral tenure at McGill. Most of those projects I had many obstacles, but this one, I don't know, for some reason everything went very smoothly because some of the infrastructure was there, and it was really the only missing piece was my system and my model. So, but the point is, the administration at our hospital, they are more than happy to leverage this in the system. But the point is, in basal cell carcinoma, 3 departments are involved. We have oncology department, we have a skin dermatology department, we have pathology department. So this is a little bit tricky to try to coordinate between these 3 departments. So the only maybe obstacle that I see is the coordination between these 3 departments. But other than that, everything for my project went very smoothly in terms of the infrastructure that I needed, and they provided. They provide because again, as I mentioned, it was coming from a biobank REB, original REB that we had, so it went very smoothly. But the coordination between these 3 departments, that sometimes they might be hostile to one another. So that's yes.
Okay. Okay, thanks. And Ali, do you have something to add?
For this project, we are still early on and we are expanding it. It's like the one that I work as part of this project was only one modality of patients. So we are basically expanding the model, and also we are collaborating with BC Cancer to use it in clinic. So this is basically a larger tool that we are over time building to give access to pathologies, at least for a pilot project, and then see how it will perform in real clinical trial or in real clinical practice. But I think the major obstacle is just it's a bit slower in clinic, using these in clinic and then trying to translate the AI part, and like there are like some skepticism toward AI still. I think that may slow down the process a bit.
Okay, thank you.
Thank you for your question.
Okay, thank you for that. Natalie. You can try again if you'd like, but I can also.
So my question was actually, it's for Ali. The results that you have are really promising in the AI section. You had mentioned that it's unsupervised model right now. And I am wondering what your plans are to refine the model going forward. Is that moving to a supervised model? How do you do that?
That's a really good question. Actually, this is what I'm working right now. So we have this AI, magic that we can train model unsupervised without any human supervision on the image side. And also we have a valuable source that are clinical information like pathology report, oncology report that are supervised like human knowledge. So what we are doing right now is basically trying to build these unsupervised image level models and then those human level knowledge models, and then try to find a mismatch between AI knowledge and also human knowledge. And that's going to give us some really important features between what AI thinks versus what human thinks. And I think that's a place that we can find where human misses some information and also where AI misses some information. This is the way that we are moving forward, but also there is this possibility that make this model supervised, but if we want to make this model supervised, we need more data. So it won't be as effective for rare cancers. The problem with rare cancer is that we don't have enough data. So going towards supervised task is not be a good solution. So these are the reasons that we try to go this way and maybe come up with new solution. And the other aspect that we are trying to move is to actually use AI to search case specific within the entire database of health data that we have publicly access to, which would be something like a ChatGPT that you ask question and goes search within the internet. We want to add that to the model too, so the model is able to search within the entire publicly available data, and maybe it finds some similar cases and give you some access to some new information.
Oh, that's really cool. And I think as the gold cohort grows, you'll probably have better opportunity, which gives us all incentive to grow that cohort.
Yes, exactly. Yes. If we have access to basically right now, a problem that I have with POG is that it is actually 24 different organs. But because lack of enough cases in different organs, we had to remove some of the organs. So meaning that if the entire Marathon of Hope cohort expands, we can actually more reliably test these models and probably give a better model to the clinic and also use it for patient care. Thank you for your question.
Okay, thank you, Ali and Natalie. I know we have a couple questions in the Q&A in the chat, but I'll call on Denis to ask the next one.
Congratulations, everybody. That was a really exciting presentation. I have a couple questions, not that I did not understand, but it kind of stimulated me to, think about cancers and maybe the future. And I'm a member of the patient working group, and one of our values is equity. So I'm going to ask a question to Riley and Peter. So everybody will have had a question today. First, Riley, one of the words you mentioned at the beginning talking about pancreatic cancer was that it was a cold cancer. Mm-hmm. And I've heard often this word, like turning a tumor into a hot tumor. Meaning like there's a migration of cells of the immune system into it, and that will help fight the cancer. So, and you mentioned different type of cells. So I had two questions, like, are there treatments now that do exactly that? Like, like they start fighting the tumor, but at the same time they call the immune system and say, hey, come on, come and help us here, And,
yeah, there's some,
And the second part is like, what's the best profile of the different cells, like kind of a signature that would be like, if I ever get another cancer again, like the signature that I'd like to see that, and I'm fighting, my immune system is fighting really hard
I'll start with the second one because that one's a little easier to understand or answer from my research. But what we saw was that when you have not only T cells, so those are the ones that can kill the cancer cell or help activate other cells, but those with B cells, so you want helper T cells, cytotoxic T cells, and B cells, at least in pancreatic, that showed the best survival rates compared to any other immune signature. And there is data in other cancers too that like a lot of people are focused on T cells, showing that they often are associated. That's why CAR T cells are therapies that exist because they can go in and kill cancer cells. So at least in pancreatic, it's those 3 cells together that's like the best signature, you could say. And I'm sure some truth to that carries over to other cancers, although it would need to be studied a little more in depth if it's like specific to each cancer. And then for the first question, there are some therapies that we call immunotherapies that, as I mentioned, boost the immune system. So things like monoclonal antibodies, what these do is they're little proteins and they go into the tumor and they can stick to a very specific, I guess, molecule in the tumor. And that makes it look more attractive to the immune system. And so there's drugs like that. There's other drugs that help turn on the immune system. To make it more active. Doesn't necessarily recruit it, but there are a bunch of different kind of options that people are going at. There's things called like the cancer vaccines, where you give the immune system, like a normal vaccine, a piece of a protein or like something on the tumor that the immune cells can look at and recognize, and then they say, oh, that's also on this tumor, let's go there. So there's a few. There's nothing that works perfectly right now. And for pancreatic, it's kind of hard because there's not really a specific marker on the tumor that makes it very attractive to the immune cells. But there are, again, studies that are working on doing that and new and upcoming targets all the time that make it more attractive to these good cells that we've seen.
Okay, thanks. Thank you. And for Peter, I'm going to say something, but your presentation was clear. It's just that I got a little bit confused. I attended the meeting in the MOHCCN network meeting in Winnipeg, and I think there they were talking about what they call circulating DNA. Like a tumor or any cell, they break down and they release DNA. And you can measure those as a way to predict cancer or, as a signal like, oh, you might have something. And you were talking about circular RNA. And so I was wondering if it's me, I did not understand well the first time around, or there are actually 2 types of things that you could measure in the blood of a regular person, like some circular or circulating DNA plus your circular RNA. And if I understand well, that could be like a signal of like the presence of a cancer or a tumor somewhere in your body. So am I right in my understanding of, well, these 2 type of, expression, I guess, circulating DNA or circular RNA, and the potential use in the future of those measurements as a way to predict or the presence of a tumor in yourself.
Yeah, that's interesting. It's so circulating tumor DNA is looking at like DNA from dead tumors that escape into our bloodstream. And so they use kind of this debris to measure the tumors, things like mutation or like if that signals things like treatment response. Where circulating RNA is different, circular RNA, sorry, is different is that it's still produced by living cells. So both normal and cancerous cells do produce these molecules as part of regulating gene expression. However, the only difference here is that they are circular in structure. So both could be explored as cancer biomarkers, but for different reasons. So things like ctDNA would tell us about the tumor's mutation through the bloodstream, whereas circular RNAs would tell us what the tumor is actively doing, since it's regulating gene expression in the living tumor cell and in normal cells as well. So that might reflect more of cancer biology, but definitely there is a need to have a panel of markers to kind of get a better idea of disease state rather than relying on a single static marker. So that could be interesting to combine both modalities as well.
Oh, okay. So this is great because, I mean, that's like what you were saying, the circular RNA is more stable. I'm not too sure about the circulating tumor DNA, but that would make it detection of cancer, if I understand right, pretty much accessible to anywhere in Canada, because anywhere in Canada, a doctor could take a blood sample and stabilize it in some ways, and then it could be analyzed by people like you, and it would be like a signal or a biomarker of sort, saying that, oh yeah, you might have potentially cancers and better start looking more in detail for detection of it and eventually for treatment. So that's pretty exciting, I'd say. And again, the patient working group, some of the value is like this technology, precision oncology, has to be accessible to anybody, no matter where they live in Canada. And very often, if you're not living close to a big research centers, well, maybe you're not as advantaged as if you would live elsewhere in Canada. So yeah, thank you for doing that type of work.
Thank you very much, Denny, for those great questions, and to Riley and Peter for the detailed answers. We do have a question in the chat from Dr. Benukraf. It's a follow-up to Andre's earlier question, so it's directed to Michael. Michael, did you account for any potential confounding factors, for example, from medical records or other external sources? I assume in terms of the differences between sexes.
Yes, of course we did. So I have clinical metadata for just about most patients since they came from a clinical trial. It is sometimes somewhat limited in the amount of information that I have. I took into account all the usual confounders that you might look at, such as stage, for example. There's a few limits here because we only have multiplexed imaging for that. So the distance measurements are based on only early stage disease, right? So you can't look at what happens at later stage disease. But in fact, the sex-specific differences came out we identified those by looking at sex as a possible confounder, because we first looked at everything and found no difference anywhere with anything. And the moment you split by sex, just as a check to make sure that sex isn't confounding things, indeed, that's exactly what we found.
Thank you very much. Oh, great. Thank you, Twitty. I had seen something from Jim in the Q&A. It's no longer there. Jim, I have allowed you to unmute yourself if you still have a question.
Sure. I think it was from Michael. I was looking at the time. That's why I deleted it. Which was that I think it was Michael who showed the Kaplan-Meier survival curve for the differences in whether or not a tumor was immune hard or not, or I can't remember exactly the what was being marked, but the difference was actually, although it was there, it was significant, but because it was 10,000 cases, it was pretty narrow. And I'm just wondering, is this because you start off with some flare-up, some inflammation in tumors, but that gets resolved, or the tumor then evolves against that, which then minimizes the overall impact over time? Or is it something, is it just because you're looking at a huge amount of data, and so there are outliers which sort of get lost in that?
Yeah, so the purpose of that figure was to sort of introduce the general audience to the role of the immune system, right? And so I wanted both a pretty figure that also speaks at the magnitude or like rather the importance across cancer types. And it was actually kind of hard to find a published figure that would be both pretty and fit my format. So what I did instead is I just looked at all of TCGA, which has the 10,000 cases, and I just looked at gene expression of CD3, which is expressed in T cells, and I split the patients into high versus low, right? And so it's averaging across a whole bunch of things. It's CD3 expression as a proxy for immune involvement. So it's not perfect, but it was just there to illustrate the point.
Yeah. Maybe Dr. Vayet would question the dependence on just measuring CD3, but that explains it. And I think also clearly immuno-oncology and immunotherapies are incredibly promising, but it's so frustrating that we don't really understand how to convert cold to hot. Coming back to one of Denise's earlier questions, and it's that would be a major breakthrough for sure.
Yes.
Thanks for that.
Yep. Thank you for that. We have a few minutes left. I have a quick question for Riley. We talked a little bit about how to make, or you talked a little bit about how tumors can be made hot, but I wonder if your data had any indication of any sort of association of why these clusters are forming. Did you find any association with expression patterns in the tumor cells, or?
Yeah, so we haven't gone to do we're going to get the sequencing data to do the RNA and DNA on this. And right now I'm looking at the genetic mutations, and I've just started assessing that. So I haven't gotten to the point where I can see associations, but I'm expecting we'll probably see some differences in probably like chemokine signatures. So like the kind of the proteins that make the cell move. I've seen that in like early results in other data sets. So I'm thinking that's something we'll see a difference in. But yeah, I'm not sure yet. The only association, there's actually no associations between like clinical factors and the immune cells, which was interesting. So yeah, I'm thinking we might see trends with mutations and probably like pretty strong associations with gene expression, but I don't know yet.
Okay, well, that's really exciting to keep an eye out for. Thank you. I also have a question for Peter. So Peter, at the beginning of your presentation, you mentioned how circular RNAs are not captured by a lot of molecular profiling methods. And I was wondering if you can talk a little bit about what is different about data from the Marathon of Hope Cancer Centers Network and why you can look at it, and if there's any, I guess, additional profiling that would be helpful in the context of your projects.
So circular RNAs don't have the typical feature, I guess in this case would be the poly A tails from linear RNAs, and a lot of protocols to sequence RNA look at essentially enriching that poly A tail to capture all of those RNA species. So in that case, it kind of doesn't take into account the species that don't have that poly A tail, so like the circular RNAs. Where the Marathon of Hope is different is that it captures total RNA, and so as such, it's able to capture those circular RNAs, and also it sequences at a deep enough sequencing length to capture those smaller populations. In terms of other modalities, I think it would be interesting to include other like whole genome sequencing and other information to kind of link the RNA expression with the gene expression as well.
Okay, great. Thank you so much. That brings us to very nearly the top of the hour. So I don't see any other questions in the chat or the Q&A. So I think we'll wrap it up here. I do want to say a huge thank you to our speakers. A few people have mentioned it, but really your presentations were fantastic. So thank you so much for the effort that you put into those. And also a huge thank you to our patient working group members, and a special shout out to those of you who were able to join the pre-meeting and participate in that exercise and help the presenters improve their presentation. So I think that those interactions were really valuable. So thank you for your participation in that. And thank you to everyone who asked questions today and participated in the seminar. I think it was really great way to start this series. So with that, happy holidays to everyone. We will do another one of these series in the new year. And we'll have a few more seminars before then too. So keep an eye out on your emails and your socials. And I hope you'll join us again. Thank you so much.
Bye for now. Happy holidays, everyone.
Thank
Thank you.